so our next speaker is Michele Gilbert from Bio-Rad Laboratories. And I wanted to open up and just show her information. And Michele originally was a chemical engineer, and then she went for a PhD in bioengineering at University of Washington. And she now is a lab manager, and she works at Bio-Rad. And you were a lab manager at Berkeley before that? Yes. All right. And with that, I'm going to turn it over to Michele. Thank you.
Thank you, Kathy, for the invitation. And since I am the one corporate, I don't work at a community college or teach, but we do work very closely with educators when we develop kits. So let me just start with this. So again, I'm at Bio-Rad Laboratories. I'm part of the Biotechnology Explorer program. And what we do as part of this program is work with educators and determine what educational needs they have in the world of biotechnology and develop kits to aid educators in teaching these concepts to students. The kit we introduced last year is called the Biofuel Enzyme Kit. We also got the front of the catalog, which is this picture here. So what I'll be talking about today then is our biofuel enzyme kit, teaching enzyme kinetics using cellulosic ethanol as a model system. The very first thing is why even teach about enzymes? Enzymes are a very core concept for chemistry, for catalysis, as well as biology and biochemistry courses, whether offered at the high school level or community college or university. As they're taught right now, typically it's just how enzyme reactions occur. It might not be a particularly interesting enzyme system for students, and the idea is to engage them in why they're learning about enzymes, and so that's why we chose biofuels. It's a very real-world application of enzymes, and in fact, the U.S. Energy Independence and Security Act of 2007 requires that by 2022, 36 billion gallons of biofuels, including 21 billion gallons of advanced biofuel or cellulosic ethanol, will be used in a replacement for gasoline. So if we can teach students an important fundamental concept such as enzymes and do that in the context of a real-world problem that they need to work on in the future, that we as a country in the world need to work on, hopefully that will engage them. The kits we develop are hands-on labs, and that also we hope engages students, is the ability to do the experimentation themselves, to control the variables and see the outcome. And these labs can also be used for teaching optimization for manufacturing, not just a basic concept of what is an enzyme. And they're highly interdisciplinary in connecting physics, chemistry, biology, and environmental sciences. So biofuels are a wide range of fuels. It's not just one thing. They're fuels, the definition is fuels that are produced from a biologic source that was recently living, so that just differentiates it from petroleum-based or carbon sources that were sequestered millions of years ago. The main sources of, or the main types of biofuels that are out there right now are biodiesel, so that's the one that a lot of people have heard of. There's the veggie diesel. You go to your McDonald's, get the used oil, refine it, and you can run your diesel vehicle with minimal modifications. There's also syngas, which is a burnt form, burning and collecting the gas and using that as a fuel source. If you look around, especially in California, where there's a lot of requirements for emissions on gasoline, you'll see may contain at least 10% ethanol on the gas tank, on the fuel pumps. And that's right now typically ethanol that's produced from starches and sugars. And that's called first-generation ethanol production. The final form of biofuel is cellulosic ethanol, which is produced in a different methodology than the starch and sugar-based. So again, first-generation ethanol production, that's in production right now. This slide is from Great Lakes Bioenergy research center. That's one of our Department of Energy sites. And so right now you can take sugarcane and immediately isolate glucose from that, ferment it, and produce ethanol. You can also start a little further upstream. Corn is a source that's used quite a bit. It has starch in it, and that starch can be isolated. You can either heat it up or use enzymes to produce the glucose and then ferment that glucose to produce ethanol. The problem, however, with using starches and sugars is right now there's a huge debate. It's called the fuel versus feed versus food debate, because your sugar canes and your starches are used for food sources, either for animals or for humans. And so if you move and shift a lot of that source over to fuel production, now your food prices go up, and that has a huge impact. And also it might change which land you use for what resources. If you convert it all over to corn or all over to sugarcane, it has a huge impact on the environment. So the push in industry is to try to use cellulose to produce ethanol. However, here's some chemistry for us. Both cellulose and starch are made up of glucose molecules. However, the difference is in the linkage between those glucose molecules. Starches are made up of alpha linkages between the 1,4 groups, the 1,4 carbon groups, whereas cellulose is made up of beta linkages. And if you think you can eat starch, you can eat spaghetti or bread and digest it. However, grass, which is a high cellulose content, we can't digest that. There's needs for, if you're a cow, you have a rumen, so you have specialized bacteria to do that. Composting bacteria are very efficient at cellulose digestion, but enzymatically it's actually a much more difficult process. However, cellulosic ethanol is a very viable source, or cellulose is a very viable source of ethanol because cellulose is the most abundant biological material on Earth. It's a huge resource. It's found in cell walls, though the cell wall composition varies because cell walls aren't only composed of cellulose, they're also composed of hemicellulose and lignin. And so we lack an efficient way to break down plant material and convert this cellulose into actual fuel. And so one of the ways, this is just a diagrammatic of how it is currently done. There are a lot of sources, depending on regionally where you are, of cellulose sources. Poplar trees are used where they grow naturally. Switchgrasses, prairie grasses are used quite a bit. Corn stover is also being investigated. That's all the bits that you don't use as kernel corn for feed or for fuel. So again, that's another advantage of cellulose as an ethanol producing compound is there's a lot of it, and it grows in different regions, so you're not just dealing with areas where you can have corn or sugar growth. But in order to get that cellulose, you first need to physically grind it out. So if it's switchgrasses or corn stover, you need to physically break it up. And once you've done that, the issue with the cellulose is it's mixed in with lignin. So in schematic B here, the cellulose is depicted as the long blue rods, and the lignin is depicted as the pink beads. Lignin is quite poisonous to enzymatic reactions, so a lot of research is being done on how to separate out that lignin from cellulose. Once, by the miracles of graphics, you get to slide C, you're now at purified cellulose, but even cellulose itself is not easy to break down. It's crystalline in structure. It has a high level of hydrogen bonding that take long strands of the glucose molecules and bind them up into multi-strand columns. So there are 3 types of enzymes that are used typically to break down that crystalline cellulose. I'll go into a little more into those in the next slide. We have C going straight to D here, but really what happens, you break down the cellulose to glucose, you still need to ferment that glucose to get ethanol, then you need to distill away the water in order to get an ethanol that you can use as a fuel source. So there's still quite a few steps between C and D. So here's my attempt at animation for cellulose breakdown. This is again assuming you have the lignin removed, and so these columns of beads are columns of glucose molecules. each row is covalently attached, and the dotted red lines are the hydrogen bonds. So with heat or acid or ammonia or other treatments, you can break apart those hydrogen bonds. And so now for simplicity, I'm just showing one particular strand. And now you can add a combination of enzymes. The combination is endocellulases, exocellulases, and cellobiase. The endocellulases break down the bonds in the center of the chain of glucose. The exos break down at the ends, and cellobiase is an enzyme that breaks down cellobiose, which is two sugar groups linked with the beta-1,4 bonds. So again, the endos and exos will go in and break in the middle bonds, and when you have just two, the cellobiase will go in. Now you've got your glucose, which you can further downstream process to ferment to ethanol. So this is just a closer look at cellobiose, which again is the two glucose sugars, and they're, for the chemists in the room, the 1,4 bonding between, or the beta bond between the 1 and 4 carbon groups in the 2. So you'd have the cellobiase enzyme come in with the presence of water, and now you've got two glucose molecules. So this is all good and well, but how are you going to teach students about enzymes with using this particular format? Cellobiose, the sugar, goes into solution nicely, but it's clear. Glucose, the product, is also clear. What we've discovered at Bio-Rad with our 15 or so years of developing educational kits is students aren't very excited about clear things going to clear things in small volumes. It's just not something that helps them learn the concept. So, and it's also very difficult to either track the disappearance of cellobiose or the appearance of glucose. There are chemical methods of detecting them, but they're not straightforward. So what we decided to do was to use an artificial substrate, which is paranitrophenyl glucoperoxide, which allows the reaction to be tracked by monitoring the appearance of a yellow color. So the top molecule you see there is cellobiose, and the bottom one is the paranitrophenyl glucoperoxide. It also serves as another purpose of a teaching point for students, because when they learn enzymes, they learn either induce FID or lock and key, which is one enzyme, one substrate. But in reality, many enzymes are actually what we would call promiscuous. If it's close enough, it's good enough, I'm happy to break it down. So in this case, it's not a beta-1,4 bond between the glucose and the nitrophenyl, but it's a similar enough bond that the enzyme can work on it.